Okay, I'm going to now go through the biotechnology section. I'm sort of going to use this video and the notes. I've actually updated these notes a little bit from the ones I gave you early in the semester, so I will give you an updated copy of those. I'll put them on the Files tab on Teams. You'll probably be the easiest way to access the videos as we go through. Now, the book divides this into two sections. It talks about biotechnology. Sorry, I'm just quickly looking at the book now. Tools and techniques, and then I think it looks at, as a separate section, applications. I don't think we need to divide them up into that too. I think it's best to handle both of those sections together. Um, so what we have here is our usual science understanding from the SACE board for biotechnology, all the things we need to know. It is important to be aware of these and use them as a checklist. What I've done is put them into a summary of what I think is more useful here at the bottom. So basically, when we're looking at biotechnology, we're going to look at a whole series of different techniques that we can use in biotechnology. And then finally, we'll just talk about some of the ethical, economic, and cultural issues associated with those at the end. So I think if we think it over as learning about all these different tools that a biotechnologist can have in their toolbox um, to do various things, procedures in biotechnology. That's probably the best way to think of it. Now, we don't actually need to know about all of these in huge amounts of detail. We just need to have a little bit of an overview of each. So briefly to introduce them, gene probes, which are used to identify specific genes, and they actually get used in a couple of these other techniques as well. We'll talk about that. The polymerized chain reaction. This is a way to copy DNA to produce large amounts of DNA. So if we have a sample from a crime scene or something like that, we have a very small sample, we need to increase the amount of DNA so that we can do an analysis of it. With some other technique, it's a way to basically photocopy the DNA, I like to think of it. DNA profiling, which is what we use for criminal cases and also identifying paternity and that sort of thing. It's also known as DNA fingerprinting. We'll talk about that. And I'll also briefly mention DNA sequencing, which is basically a bit like profiling, but it's a much more comprehensive method and will tell you the order of basically every base within a gene. We'll talk about how we can transfer genes from one organism to another or one species to another. We'll talk about transgenic organisms. When we get to that, I'll talk exactly about what they are, but you might be surprised that you're already familiar with some of those. We'll talk about cloning, one that's not so much in the news now, but has been in the news. We'll talk about a new technique called CRISPR, which has got some very, very exciting possible uses and applications. And as I said, finally, the ethical, economic, and cultural issues. So as we go through this, think about, I've got to have a rough idea about 1, 2, 3, 4, 5, 6, 7, 8 of these different tools, and, pardon me, and some of the ethical, economic, and cultural issues associated with those. So let's get into gene probes. Now, I strongly recommend at this point that you pause this video in a separate window, maybe just link to it from your notes. So if you open up the notes and just do a control click on this link here and just watch from 0 at the start to 1 minute 15, and this video will run you through a really good idea of what gene probes are. Just hopefully you've paused, watch that now, come back, and now you're still watching this video. So just to summarize what we've got in the notes, what we do when we probe for DNA is we produce DNA probe, which contains two things. It contains a complementary nucleotide sequence to match the target DNA in our target gene. So we're trying to find a certain gene. We build a probe that has the complementary strand that will bind onto that. Then we attach a radioactive or fluorescent label that can be readily identified. So then when we add that to our DNA, if it attaches, we can clearly identify where it is or if it's attached because it's got basically this marker on it that's either a fluorescent molecule or a radioactive molecule that we can easily detect with something like an X-ray some other technique that can detect where that probe DNA is. To do this process, basically, we need to do what's called hybridizing the DNA, which is just a fancy way of saying we heat it up to separate it into strands. So we start with our sample, we heat it up, it forms two strands. We add these probes, which have these labels on them, either radioactive or fluorescent, a molecular beacon they call it here. Because these strands are separated, these probes, if they find the target gene with the complementary bases, will attach to that, and then we are able to quickly identify if the target gene is present within a sample. After we've identified that gene, we may want to remove it for some reason, and we do this with something called a restriction enzyme. So these restriction enzymes are very important, talked about them in the video, and they have the ability to chop DNA into sections, and potentially then therefore we can chop out our target gene. So just briefly here, how are restriction enzymes used in the manipulation of DNA? Restriction enzymes are used to cut DNA into smaller fragments. Different restriction enzymes recognize and cut different DNA sequences at specific nucleotide sequences. So basically what that means is at the end of each of our genes, we have a little sort of characteristic set of bases, which is basically a marker for the end and the start of a gene. And a restriction enzyme has the ability to go in, find those markers, and basically cut the gene at that markers. And it cuts it in a special way so that we get these sticky ends so we can reasonably easily join it to something else. And we'll talk about that when we talk about one of the other techniques later on. PCR, polymerase chain reaction. This is a process we use to basically make lots and lots and lots of copies of a gene or a segment of DNA. So as I said before, might be one that's collected from a crime scene or something like that. It's not enough to do a proper analysis, so we can go through this process to copy it and make large amounts. And what it basically involves is, firstly, we heat the DNA again to separate it into the two strands. That gives enough energy to break these weak hydrogen bonds between the strands. We then add primers, and these primers are basically just small sections of, if you like, DNA, or small, a small sequence of nucleotides. These new nucleotides attach to these two new separate strands, so it's a little bit like the DNA replication process. Then we, after we've added the nucleotides, we add a DNA polymerase enzyme, which basically does the same thing to in DNA replication and runs along and zips all those nucleotides together to make a new strand. Then, after we've done that, so now we've gone from having one strand, we've built two strands now, like in DNA replication. Then we'll do that for a second cycle, and we will go from 2 to 4. And then a third cycle will go from 4 to 8. And we might do this up to 30 or 40 cycles, and we can go from having a very, very small amount of DNA to making lots and lots and lots of DNA, so that we can go and do some other analysis on it. As I said, if you haven't already, pause this video and watch both these videos. This first one's quite short, and both these videos go through and explain the process of the polymerase chain reaction. So if you haven't paused already, pause now, watch those videos, then come back to me and I'll be talking about the next section. Okay, so the next tool is DNA profiling. This is the one you see on all the TV shows and the criminal TV shows and all that sort of thing. Or, when we want to find out, is our dad really our dad? Not that I've ever had to do that, and I hope no one else has, but it is used for that. So, probably best pause the video now, have a look at both these videos that go through the process of DNA profiling. I think they explain it probably much better than I can because they use much better graphics. Have a look at those, and then I'll come back and I'll just briefly talk you through my notes so you can see that hopefully they make sense and relate to what you've just seen here. So the first thing that needs to be done to DNA profile is to extract the DNA from the cells. That's a similar process to what we did with extracting the DNA from the P cells, either in earlier years, or I did demo that briefly for the guys at Streaky when I was there recently. Secondly, if necessary, which is often the case I talked about before, if we need more of it, then we need to do the polymerase chain reaction to increase the amount of DNA. Then all these things are coming together with the PCR. Now we're going to use restriction enzymes. And what the restriction enzymes will do is they will cut the gene up into fragments. Now, everyone has unique DNA, unless you're an identical twin. Everyone's DNA breaks into different fragments. So the fragments that my DNA will break into when it's cut up will be different to the fragments that maybe Cabe's DNA will break into when it gets cut up. So then we use a process called gel electrophoresis. And this basically separates the fragments based on their size. So we basically start with a agarose and buffer solution that's poured into a plastic tray. The comb is placed onto the plastic tray at one end, here like this. The agarose polymerizes into a gel as it cools. The comb is then removed from the gel to form wells for the sample. So we get all little holes here where this comb was. If you've ever done agar plating, it sort of forms like that. It was basically like a hard jelly if you've ever made jelly forming. We then introduce the DNA into each of these little holes from where the comb was, and we have a dye attached to it so we can track where it goes to. Then the tray is placed in a chamber that generates electric current through the gel. The negative electrode is placed on the side nearest the samples, up here, and the positive electrode is placed down here. DNA has a negative charge and will be drawn to the positive. So DNA is negatively charged, so we get repelled from the negative, attracted to the positive. Don't need to know the real detail here, just have a familiarity with how it works. And basically, the smaller a fragment is, the faster it'll go. So a small fragment will travel right down to here, larger fragments will travel up to here. That will then basically leave this characteristic, what we call fingerprint. So this pattern of how many fragments of DNA you have of different sizes, because everyone's DNA is different, everyone's will look different, and then that gives us basically this like characteristic pattern or fingerprint of our DNA, which is then obviously can be used to help identify say a suspect in a criminal if we check some DNA that was taken with them, with some stuff that was maybe found at a crime scene. Now with paternity testing, it's a bit different. My DNA will be similar to my father. It won't be exactly the same, but it will be similar, and my mother. So you will see similar fragments, similar patterns in the fragments. They won't be exactly the same, but a number of the fragments will be similar to my father and a number will be similar to my mother. So by looking at their DNA and comparing it, we can tell whether those two match up. If the pattern is completely different, that would mean that we are not probably closely related and that maybe my father wouldn't be my father.